I was gonna make my wife a cake, but I was too lazy. Oh, I'm sorry you're feeling glum, Jay. Hopefully you'll turn that around pretty soon as school ends, not too soon from now. Okay, so as you can see, we're continuing on with our unit. I should have put it on here. I didn't. I forgot to. You know, I'm going to do it right now while I'm talking. If you don't remember, I mentioned on Friday last week that our unit exam is coming up on Friday. Put it here. I know it seems like we just had a test, but believe it or not, that was a couple of weeks ago. So Unit 3 is going to be like everything we've been talking about from biomolecules up to now. So the biomolecules, cell cycle, mitosis, which is what we're talking about today, all that stuff is going to be on the test on Friday. So again, I know it seems like it's just happened, but that's just because the days are going by so fast. It's already November. So don't forget our exam coming up on Friday. If you didn't have a chance to copy down the objective, you'll get a chance here in a little bit after the do now. Notice our vocabulary. Got some new words up here, as well as new key points. And we're going to go ahead. Oh, one more thing. Core value of the month, no excuses, right? It's a new month, new value, so no excuses. We've got lots of vacations coming up. We've got our Thanksgiving break in about 3 weeks. And then after another couple weeks after that, we'll have our winter break. So we've got lots of stuff happening. The year is like almost halfway over, if you can believe it. We'll be having a mock star sometime pretty soon. I'll have more info for that for you guys coming up as I learn more. But just keep that in mind that we'll have our mock star pretty soon by the end of the semester. Okay, here is our do now. There you go. Here is our do now. Let's go. Well, thank you, Cheyenne, for getting a picture of that. And Tatiana, the reason the schools are closed would be so that parents and teachers can go vote. Because as it is, a teacher who's teaching from, we have to get here before 7, we don't leave before 5. That's, for the most part, polling locations are open during that time. So we'd all be rushing to get to the polls and kind of a big mess. We leave around here around 5. But school for you guys is over like at 3:20. We're not closed, but other schools, according to Tatiana, are. I've not heard that, but I'm glad to hear it. Let's see, Kaylin had family from Corpus Christi. That's nice. It has been light years, Casey. Dog years. Hello, Kaylee. Hi, Zaria. Thank you, Miguel, first. Oh, no, Casey's first to respond. Miguel and Casey. I will admit number 2 is a little bit difficult, so don't rush through it. Take your time.
Mr. Martinez, you have to give me a minute on number 2, because that's going to take a long time to explain.
Okay.
Is this to do now?
It is.
Okay.
Hi, Gigi. Thank you, Karina. Thank you, Grace. Like I said, that number 2 is no joke. That one's a little bit harder. But I can see you guys are doing really good with the DNA. Glad to see it. Remember, those are easy points on the star. Easy points. Thank you, Brandon. Remember, don't forget the number 2, a little bit harder, a little longer. And also, for those who might not remember, I am taking your chat responses as a part of your daily grade. Thank you, Faye. So keep that in mind. I download the chat box after we're done. I go through there, make sure everybody's answering.
So, Mr. Martinez, how was your weekend?
It was all right. How was yours?
Pretty all right. So, let me guess, you had to do papers on the weekend?
Sorry, one more time. What was that, Brandon?
I said, did you have to print out some papers on the weekend?
Print out some papers. I did not. But why would I do that?
I mean like paperwork, something like that.
No, I had a pretty chill weekend. Mostly just watched spooky movies with my family and ate a bunch of food. Ooh, Brighton. I think no, Gigi might have been the first. I'm gonna give y'all one more minute. Let's go one more minute on our warm up because I want to make sure that you at least attempt the written answer. So if you haven't tried number 2 yet, make sure that you do. Again, this is for a grade. Thank you, Gigi. Thank you, Brighton. Thank you, Davian. Oh, I think I might have been a little too hard. A lot of you are struggling with this one. Josiah, I'm reading your answer. It looks really good so far. Okay, Miguel, we'll talk about the DNA strand here in a second. Miguel Gallegos. Okay, I did see some good responses. I didn't get a chance to read all of Zaria's. Zaria, would you mind coming off of mute and reading your answer for me?
Okay, so I put you want me to read like the second answer?
Yeah, let's go. Everybody seems to know what they're doing on number 1. Let's go to number 2.
Okay. So I put right after the cell divides, it goes into a G0 stage where it rests and regains like strength, I guess. And then the first actual process of it is G1 stage, is where the cell blocks off any disformed or unregulated cells that could be like like that could be a problem to the future cells, like cancer and stuff.
Right.
And it does this because in the S stage, S stage actually is where the cell begins to build organelles and actually make the cells for the DNA replication. G2 is where the DNA replication happens, where the you know everything in the cell basically, and
Very good.
then in the interface is where mitosis happens, and that's when the cell actually divides and make a whole new cell.
All right, I want to correct you on one thing because interphase is going to be G1S and G2, whereas mitosis is not a part of interphase.
Okay.
But aside from that, very, very good. Thank you.
Welcome.
And I saw a couple more. Cameron, can you come off mute and talk about yours? No luck. That's fine. No, I couldn't hear you, but that's okay. We're going to move on anyway. So here's what I put for number 2. Oops, sorry. Well, I guess first we'll talk about number 1. So if you still don't understand the binding rules, we'll talk more about this during intervention this week, because I think at this point, pretty much everybody understands this, right? A to T, C to G. So if you didn't get a chance to, here's the answer that you can double check your work from. But okay, let's talk about number 2. So here's what I put for number 2. I know, I guess looking back now, maybe put too many vocab words for you to use, but what I really was looking for was for you to really summarize the cell cycle. So the cell cycle is made up of 5 phases, which are G1, G0, S, G2, which is interphase, and then the M phase. So again, remember that G1S and G2 are separate from mitosis, right? G1S and G2 are interphase versus M, which is the mitosis. Now, DNA replication happens during the S phase of interphase when the DNA is duplicated while the cell grows during the G phases. So again, during the interphase, those G phases are when the cell grows, and the S phase is when the DNA replicates to get ready for mitosis. Cancer results from uncontrolled or unregulated cell division. So there we go. A lot of words, probably looking back, too many words. But the point is, this is how a cell grows and divides, right? Is through the cell cycle. Are there any questions about that process or about this thing going on? No questions? Okay, then let's continue on. So, can I get someone to read the objective for today?
I can read it.
Go for it.
Students will be able to describe the phases of the cell cycle.
Very good. So we've been talking about specifically the cell cycle, and now we're going to focus in on mitosis. So here you can kind of see different cells and different phases of mitosis. All right, so here's from last week. I wanted to show you this one more time so that you could see it. The one on the left is like a microscope. This is something that's happening like in real time, more or less, in real life, versus the comic on the right, which is like a visual diagram of the different stages. So by the end of today, you will have a really good understanding of how mitosis works and be able to recognize all these different stages. Okay, so if you haven't yet, go ahead and pull out your independent practice because we're going to answer a lot of these questions. It will take some time for you to work on it, so even after I'm finished talking, you'll have time to practice it during our independent time. But for the time being, go ahead and get it ready so that you can start taking notes on it. Alrighty. So, can I get someone to come off mute and read this for me, please? Go for it, Brandon.
The 3 main reasons monocytes
Yes.
are cures are repair of cells, development, and growth.
Thank you. All right, can I get Brian? Would you mind telling me what the 3 main reasons of mitosis are?
Hello, can you hear me?
I can hear you.
Alright. Wait, what do you want me to do then? Again, because my computer was lagging a lot.
It's all
And I
right. Tell
can-
me the 3 main reasons mitosis occurs.
Come on, Brian.
Because it repel. Oh my god, I can't read English. Repair of cells and development and growth.
Very good. So
That-
the reasons that a cell will undergo mitosis is because to repair the cell, to develop and grow. So like we're talking about with a baby going to an adult, right? That doesn't happen overnight. That happens over a long period of time. Those cells are constantly having to change and adapt and become individualized, become specialized. So that's all carried out through mitosis. So this is taken from the PowerPoint that's already uploaded into files. So you have access to this. I just pulled out a couple of slides. I don't have time to go through everything, but I wanted to make sure that I did talk about some things. Okay, so here we go with the cell cycle. We're now at the M phase. Last week we talked about G1S and G2, which are the interphase. Now we're moving into the M phase. So can someone tell me what is the defining trait of the M phase? And I'll give you a hint, it's right here on the slide.
Is it mitosis?
It is, and what's the important part about mitosis?
The cell divides into two identical cells.
You got it, man, you're a genius, right? So the is that we have cells that are dividing and creating clones. Now, we'll talk more about cell specialization later, like how those cells become more individualized, because of course, not all cells are going to be the same. But when it comes to replacing or growing or developing existing cells, those are going to be clones. So the example I mentioned last week was like when I'm picking my nails, like I said, I got a bad habit of picking my nails. You want to make sure that the cells that are growing are going to be the same cells that used to be there, right? You don't want for all of a sudden there to be like a muscle cell on top of your skin, right? Because that's where bad things start to happen. When cells kind of do things they're not supposed to be doing. That's where cancer comes from, right? Okay. Now, here is mitosis. These pictures look pretty crazy, but these are all pulled from actual cells. That's how we know this thing is going on, is because we've seen it under a microscope. So can I get someone to read this slide for me, please?
I can read it.
Go for it.
All right. Mitosis is the division of the nucleus. Only occurs in eukaryotes, has 4 stages, and doesn't occur in some cells such as brain cells.
Very good. Thank you, Hamza. So who have I not bothered in a while? Oh, Hugo. Would you expect to see mitosis happening in a bacteria cell, Hugo?
No, because it doesn't have a nucleus, right?
Very good. And it's also not a eukaryote, right? It's a prokaryote. Thank you, Hugo. So, mitosis, we're dealing with the division of a nucleus. That's why we're seeing all these crazy looking pictures, because it's the DNA that's being split in half. And because of that, it's going to look pretty interesting. Has 4 stages, we'll talk about here in a second, and it does not occur in some cells, such as brain cells. So for those of you who didn't know, you don't regenerate brain cells. You're pretty much born with what you got. So whenever you're like losing sleep or you're going out binge drinking, whatever, when you're killing your brain cells, you are not getting those brain cells back. Those are gone forever. There is some evidence to show that you can stimulate neurons to create new pathways, which is how you form memory. But as far as the amount of brain cells you have, that is finite. You don't get more of those. So like I mentioned with my finger, right? When I start picking up my finger and I start getting rid of those skin cells, new skin cells will grow in its place. What happens with brain cells is that if you lose brain cells, they just die. Nothing else happens. A big part of that is because we're going to find out here when we talk about mitosis, there's a specific organelle responsible for helping the cell divide that those brain cells do not have. Okay, so let's jump in then with...
Sir, how do you
go
lose
ahead.
brain cells? Like from
So
drink?
the 2 most common are going to be, like I mentioned, with binge drinking. So if you're drinking a whole lot, you lose brain cells. What happens is, like, you've seen like in TV shows, because I hope you haven't experienced it firsthand. Whenever you drink a lot, you have like that hangover in the morning, right?
Yeah.
That hangover feeling is literally the loss of brain cells. What happens is there's something called the blood-brain barrier up inside your head that kind of protects your brain from a lot of like dangerous things. It's supposed to work because for the most part, you're encountering things that are normal. When you start drinking alcohol, you are ingesting a chemical, right? It's not like you're drinking water. You're ingesting something that's not meant to be ingested, and you're literally doing. The side effect is there's this slight, there's a loosening of inhibitions and sometimes even a positive feeling associated with it because, like I said, you're killing your brain cells. So that death is what is feeling good. And then, of course, when you have the hangover in the morning is because of the brain damage that you have done. But it's not just drinking. It's also lack of sleep, diet, all sorts of things play into that. Oh, especially drugs, especially cocaine. Yes?
Okay. Thanks.
Miss
Yeah, you start to
Yes.
lose your memories. That's why you see things on TV where people are kind of like forgetful. Like a big thing with alcoholics is that they don't remember a lot of things, and that's because of the amount of brain damage that they have.
Wait, sir.
Uh-huh.
If you lose a lot of brain cells, is that how people get Alzheimer's or no?
It can result in that, but that's not super common. What happens is that you just lose short-term memory. When we're talking about like dementia and Alzheimer's, that's a whole different kind of process. You know how well, like when you see like fat on a stove and how it turns into that white gunky stuff? A similar process happens in your brain with certain cells that cause blockages. And those blockages prevent those pathways from forming, which is how you can get dementia. It's because it's a lot more complicated than just the cells dying. It also happens, but it's a whole different kind of process.
Yeah.
So Gigi, you're absolutely right. Force yourself to sleep. I wish that I had thought about it more when I was in college. College, I pulled all-nighters, I did crazy stuff, and I'm sure because of that, I'm going to have a lot more memory problems in the future. So yeah,
Wait,
please,
sir,
please, please go to sleep. Yeah, Brian.
is there a way how you recommend how to fix your sleep schedule? Because I have mine really messed up right now.
The best thing you can do is exercise. What the best thing you can do is like in the morning when you wake up, your body is primed, like your metabolism at its highest. And that's partially because, like I mentioned a while ago, we're designed to be like cavemen. So you wake up in the morning, you do a bunch of exercise, and that metabolism carries you through the rest of the day so you can eat a bunch of food, and then that prompts you to go to sleep once the sun goes down. So ideally, you should be getting around 7 to 8 hours of sleep a day, and no more, because a lot of sleep can also be bad for you.
Oh, okay. Thank you.
You're welcome. So yeah, exercise is key. It's harder to go to sleep when you're, playing video games all night or doing a bunch of things that are not conducive to a healthy lifestyle. But anyway, I'm getting off track. Okay, so the 4 phases of mitosis: prophase, metaphase, anaphase, telophase. And I'm actually going to stop here real quick because I wanted to teach y'all something. So there is something called a hand jive that goes with mitosis to help you remember. Again, it's one of those really silly things, but I find that the silly things help me to remember some of this stuff. So what you do is you take a fist, cover with your other hand, and you can kind of see how your fingers are spread out. This is prophase. During prophase, the cell membrane or the nuclear membrane is breaking down, so that's why it's not that's why it's not like this, it's like this. So prophase, and then metaphase when the chromosomes all line up along the middle. Anaphase when it pulls apart, and then telophase, got 2 new cells. So prophase, metaphase, anaphase, telophase. I'm like a kinesthetic audio learner, so like I need to like be able to interact with information. I have a really hard time just like looking at a picture and getting it. So this is a really helpful way to kind of remember what's going on. So it helps you internalize the information. So you got prophase, metaphase, anaphase, and telophase. Now, the reason it's important to have that hand jive is because when it comes to STAR, they're going to show you mitosis in a lot of different ways. And it's never 100% sure what way they're going to show you. It could be like a diagram. It could be like this, where they're showing you microscope pictures. So it's good to have that visual memory of like what it looks like. And the best way for me to do that is through like I showed you, that little hand jive thing. Okay, so prophase, metaphase, anaphase, telophase. Now, in the chat box, what are the 4 stages of mitosis in order? So again, your chat box messages are a part of your daily grade. I'm looking for the 4 stages of mitosis in order. Let me see what y'all got in the chat box. You got it, Casey. Good job. Thank you, Brighton. Thank you, Hamza. Thank you, Faye. If y'all are having trouble trying to think of what it is, you've already got lots of help in the chat box. Again, this is part of your daily grade. Easy points. Don't lose them. Very good. Very good. Thank you, Josiah. Jacob, taking it easy. I like it.
Stage 1, it would be the cell grows.
Well,
Number
Brandon, I
2,
think you might be getting confused with the whole cell cycle. We're looking specifically at the M phase, which is mitosis.
Oh.
So if you didn't see Jay's earlier, there's an easy way to remember it. It's called PMAT. So prophase, metaphase, anaphase, telophase. All right, very good. Thank you, guys.
Is it RATCCTAEC2?
No, it's
Number one.
PMAT, prophase, metaphase, anaphase, telophase.
Oh.
Okay, so let's talk about the individual phases. When it comes to the star, I don't expect them to ask you a whole lot of questions about the stages individually. Like I said, when it really comes to star, they're going to want you to know like what a picture looks like. Like, look at this picture. Tell me what.
Sorry for the interruption. Angel Arzate going home, please. Angel Arzate going home.
That's you, buddy. They're going to expect you to look at a picture and be able to identify the stage. Now, prophase is very hard to identify because it's like the very beginning of the cycle. The DNA is kind of condensing from chromatin into that chromosome, which I'll explain here in a second. But yeah, it's kind of hard to identify. The chromosomes appear condensed and the nuclear envelope is not apparent. So what happens is, remember, there's that double membrane. So the membrane around the nucleus starts to kind of disintegrate. That way the DNA can actually be split in half and cell can get ready for division. So when I say condense, what that means is we have that like chromatin, like the DNA is all kind of like spaghetti. There's no order to it because it doesn't really have to be in order when it's just living its life during interphase. When it comes time to divide, though, it has to condense and form those chromosomes. So when you see those X's, that's what that is. The DNA has gone from being like spaghetti into nice form shapes. And you might have heard that there's like 23 pairs of chromosomes, 46 individual chromosomes. That's what those are, those X's. So it's just DNA that's been formed into those X's so that when it comes time to divide, everything can divide evenly. Because it's really hard to divide spaghetti evenly, right? You can cut it right down the middle, that's fine, but you're not going to get an even split. The important part about mitosis is that we're creating what kind of cells? It starts with an I. What kind of cells are we creating with mitosis?
Interphase?
No, not interphase. I think I heard Zaria say it.
Zaria?
Identical.
Identical cells. Very good. Thank you, Zaria. So the reason we want that DNA to be in chromosome form is so that we can divide everything evenly. Okay, so metaphase then, like I said, very easy to identify. All of the chromosomes are aligned along the middle. So the DNA is condensed. It's in the chromosome form. It's all along the middle. And then after that, anaphase, where it pulls apart. Now, we'll talk more about chromatids and chromosomes and all that stuff when we get into genetics. For now, we're just kind of looking at the phases. So don't get too tied up on those words quite yet. So metaphase lines up along the middle, right? Metaphase middle. Anaphase pulls it apart. And then finally, we have telophase, which is where you're getting those two cells. Again, telophase is one of the harder ones to see because the cell just finished dividing. And so it's like kind of hard to tell. But anaphase, easy to identify. Metaphase, very easy to identify. So those are easy points. Don't forget those. Metaphase along the middle. Anaphase pulling it to the opposite ends. Okay. Cytokinesis then is the actual splitting between the two cells. So telophase is that last stage. Cytokinesis then is when the cytoplasm literally separates from itself to form those two cells. I don't think I've ever seen cytokinesis on a star, but you never know. They could always ask. So again, like I said, the important thing, we're producing identical daughter cells. We duplicated the DNA during S phase. That way, when we get to the mitosis, when that cell splits in half, we're not getting half of the amount of information. We're getting equal amounts. That's why we have the pairs, like I mentioned a second ago. So the chromosomes have all condensed, have all formed. They replicate during S phase, and then during mitosis, when they split apart, everybody gets one copy of everything. Alrighty. So here is a little GIF kind of showing you how that works in action. See, so during prophase, everything kind of condenses. The envelope goes away. The centrioles move to the opposite poles. The chromosomes line up along the middle. They are pulled apart. And then the membrane reforms, and you have two new cells. So it's a very, very kind of quick process like I showed you earlier. You can identify them a little bit. So here we have prophase, right, with the membrane going away. Then when everything lines up along the middle, that's metaphase. Then you pull everything apart, that's anaphase. And then when you have two new cells, that's telophase. Okay. Oof. All right, so one more time. In the chat box, correctly identify the following cell stages. You got 1 through 5. Go ahead and let me know, what are those stages of the cell's, of the cell's life. 1 through 5. Now 4 of them you already know, so one of them you have to kind of figure out what is that. So again, I'll give you a hint. 2 through 5, you already have the answers in the chat box from what you put a second ago. What I'm looking for then is the difference between number 1 and then 2 through 5. Casey is kind of right. I would prefer something a little broader than S phase. I wasn't trying to be that tricky. Again, you're still being very specific. It is a word, and more specifically, it is a phase of the cell cycle that is not M phase. There you go, Gigi. Good job. Thank you, Brighton. So when, oh, actually, you're almost there. One of those is wrong. And Josiah, if you heard, we just kind of figured out what number 1 was. There you go. Gigi's giving you the answer. So interphase is going to be that first one. Like I said, when the cell is not dividing, like I said last week, it's in interphase, be it G1, S, or G2. It's kind of hard to tell if that's G1, S, or G2, so that's why I wasn't worried about you figuring out that. But number 1 is going to be interphase, and then 2 through 5 is mitosis. So 2 is prophase, 3 metaphase, right, everything aligned along the middle, 4 being pulled apart, that's anaphase, and then 5, you got 2 cells, that's telophase. So again, number 1, interphase. Number 2, prophase, you can tell because of the nuclear membrane disintegrating. Number 3, everything's lined up along the middle, metaphase. Everything's pulled apart in number 4, so anaphase. And then 5, telophase. Very good. I know there's a lot of vocab. That's why we're trying to make sure that we're getting all these checked for understandings because, again, biology is a very vocab-heavy subject. There we go. Do we have any questions about this or the stages of mitosis? I will say that you have a lot of resources. You have the PowerPoint, which goes into more depth with all the different stages of the cell cycle, especially here in mitosis. For the most part, it's not super important for the star, at least not at this point. There are some things you'll need to know later, but we're not going to get to that until much later. But if you want to get into more information, the assignment that you have pulled up, there's a document with a lot of explanations of the different cycles and the different phases. And of course, the PowerPoint has already been given to you in files, so you can go back and review that. Okay, now, when it comes to your document about the different phases, I want to encourage you to draw diagrams. I know that it's not the easiest thing to do, but I promise you, like I said a second ago, when you try and make connections with this information by doing it yourself, you learn so much more. So I did these on AutoDraw. And in case you don't believe me, I think I went ahead and left it up. Yeah, look, I made my diagrams on AutoDraw. Oops, not that. I know that it's not going to be the easiest thing to do, but it can be done. What I want you to do then is to make these diagrams because I promise it'll help you to remember these stages. I know right now you're thinking that it's just like, you know, how hard could it be? It's just prophase, metaphase, anaphase, telophase. But when you take the star, it's going to be like, you know, after a whole year has passed and you can't remember what is and what isn't stuff, it's going to be really difficult to kind of keep all that in your brain. But if you have taken time to make your own notes, to make your own diagrams, you'll definitely remember stuff a lot easier. Well, you get the point, right? Okay, so it will take some doing depending on how you want to do this.
Wait a
If
minute,
you want to do it,
do we do the independent practice now?
yes.
Okay.
So if you want to do it on paper, you definitely can. I'm not requiring you to like use auto draw. It's just for those of you who feel like drawing is not your strong suit. Auto draw is, he just left. Auto draw is an option for you to kind of explore that without having to put pencil to paper. Because I know some of you are, you know, don't have a lot of confidence in your artistic abilities. So here, just drawing some shapes, not difficult. Again, this is not an art class. I'm not critiquing your ability to draw. I just want you to be able to make those connections. Again, if you have the PowerPoint, which you should in files, you have lots of other diagrams you can look at and reference. And again, you can always draw your own. If you just want to copy those straight from the PowerPoint, you definitely can. I'm just going to require and encourage you to do it on your own so that you can make those connections. Okay, so I'm going to give you about 15 minutes to work on this. Again, all the information you need is in the PowerPoint and especially in the document that's provided along in the assignment. All the information is there. There's no need to Google anything. Again, I'm not saying you can't Google. You can Google all you want, but whenever you Google stuff, you're going to come across things you don't understand, I promise you. The things that I've given you are a little bit easier to understand and digest, and it's really all the information you need to know. There's nothing extra. Okay? All right, then I'm gonna put 15 minutes on the clock. And here we go. Sorry?
You're delivering homework?
Oh, okay.
How many, just 5?
Yes, 5. Thank you.
You the teacher?
Yes.
Want a bath?
Sure, I'll take one. Thank you so much.
Yeah, you know it.
I have no idea what this.
Wait, Mr. Martinez,
Yes.
how would you paste your drawing?
I use, there's something that you can do on Windows computers, the snipping tool. If you don't have the snipping tool on a Windows computer, you can always use screen capture on a Mac. Or if you're using AutoDraw, you can just save it. I think that's an option as well.
I'll just draw it on paper and attach it to the assignment.
That works.
Casey. Okay, so what I do is I draw it and then I screenshot it and I crop it. I crop picture and then paste that picture to the assignment.
Are you doing it on your phone or on your computer?
On the
Thank you.
You're welcome.
Thank you, Zaria.
Yeah, you can take this off. They're like butter and salt. What's this? I think you have to shake it. Because there's like salt in there. Ah, yes, yeah. No, you know, they're really good. They do. Alright, first that was good. I don't know, but you have to keep leaving them.
Hey, sir.
Uh-huh.
Is it okay if we upload a picture from the web? Or
I would like for you to make it yourself. So I don't want you to copy and paste something that's already created. I want you to make something.
okay.
So if you don't use AutoDraw, you can always just draw it on your own, like on a piece of paper. How you're gonna do it, totally up to you. But again, I do encourage you, and again, require you to create the drawing yourself.
Wait, so what are we doing? We're just doing the independent practice, or are we drawing something?
The independent practice requires some drawing, Alan.
Okay.
Cool. I think we're the better couple. Let's get to the bottom of the video.
You know what, Avery? I'm okay with that. I will give you extra credit if you color.
Wait, so what are we drawing, sir?
So you'll be drawing for the most part cell diagrams of prophase, metaphase, anaphase, and telophase, and one of interphase. So the independent practice requires you to describe what's going on in those different phases, but I also want to see a picture of what you understand is going on.
So we're drawing a picture of a metaphase.
Yeah, so all in all, you
Anaphase.
should have like 5. So there'll be 4 for mitosis and then 1 interphase.
Okay.
You go down the hill and there's like a place like a cell phone. Inside the apartment. No? No. I thought it was only elementary then.
I know it's a pain, but like I said, it will definitely help you remember. I have posted the exit ticket. I double checked this time because of what happened last week. It is there. I'll go ahead and post it here in about 4 minutes. Actually, what time is it? I'll go ahead and post it now. There you go. So if you have finished, you can go ahead and start working on the exit ticket. If you have not, totally fine. But please get those done as soon as you can. The exit ticket again is due at 3 p.m. So make sure you're getting that turned in before you log off today. Real quick, you're not going to intervention today. After class, you'll be heading to your dens. And then from dens, we should be dismissing to the senior something or other. I forgot what it was called. Senior parade. So this is to celebrate the seniors who made the November 1st application goal. So there'll be a live stream.
To college?
Yes, to college, because it'll be for the seniors. So we're going to celebrate
Sir,
with
do we
them.
have to go?
Yeah, so when you're in den, that'll be what's broadcast. So again, not going to intervention, you'll be going to den after this. If you're here, you got popcorn to enjoy while you watch the seniors. Those of you at home, I encourage you, if you got some, go ahead and make some.
I have a cool movie popcorn machine, so yeah, I got some ready.
That is awesome. I am super jealous. One of the things I probably miss the most about the old world is going to the movies and getting some of that fresh popcorn.
Mm-hmm.
Sir, I don't know how to draw.
That's why I included auto draw. So if you feel like you don't want to use pen and paper, sorry, you can use auto draw. I included the link in the assignment. It's basically just drawing shapes. So you don't have to worry about like drawing anything that looks true to life. It's just a bunch of circles, squares, and X's. And again, the reason I'm making you do this is because when it comes to the star, I guarantee you there will be pictures where they'll show you part of the mitosis and expect you to know which of the 4 phases it is. And hint, hint, on the test, there will also be questions like that. So it's important to know which one of those images refers to prophase, metaphase, anaphase, or telophase. The best way to do that, I think, is to draw them yourself.
Do we have to do it? We have to do it, right?
Yes, it is part of the assignment. For the next couple of units, we'll be doing a lot of drawing, so probably up until February of next year, I would expect for you to be doing a lot of drawing. Again, we're not doing anything still lives or, lady by the lake or a chair thing that you have to fold, nothing like that. Everything that we draw is very simple. But I do think that it helps you to remember things. So as long as we're talking about DNA, which we'll be talking about for a long time, we'll be doing drawings because I think it's the best way to remember. I think we work on that for another minute, and then we're going to move into the exit ticket. Let's see, Kaylin. It's okay. It's not that time, so I'm sure it's fine. I was trying to remember the things that Miss Cantu has had you draw. I know they all did the chair thing, and I feel like y'all had to draw some kind of like portrait thing a while back.
Oh, our name.
Yeah, your name. Okay, so if you have not, please go ahead and move over to the exit ticket. It is thankfully not that difficult if you have notes or if you're paying attention, which I hope you were. But yeah, take a few minutes, go ahead, go to the exit ticket. Bloop, bloop. .2. It is, it's so easy. And again, if you have finished, you'll be heading to your dens for the senior parade.
Mr. Martinez, middle and center are the same thing, right?
Yes.
Okay.
And I'm going to go ahead and stop recording.